In the world of pharmaceutical and biotech manufacturing, consistency and reliability in testing are critical. That's where Working Reference Standards, or WRS, come into play. A Working Reference Standard is a validated material used routinely in analytical testing. It's derived from a certified primary reference standard and ensures uniformity in evaluating drug identity, strength, quality, and purity. Before it can be used, the Working Reference Standard must be properly qualified. Without this step, you risk analytical variability, regulatory citations, and even compromised patient safety. A well-documented qualification protocol confirms the material's suitability for ongoing use. A robust Working Reference Standard qualification protocol should cover the following elements. Objective: define the purpose of the standard. Material source: document origin, lot number, and expiration. Test methods: use validated analytical procedures. Comparative testing: analyze Working Reference Standards side by side with the primary standard. Acceptance criteria: set limits for potency, purity, and identity. Stability studies: establish retest intervals. Documentation: archive all records and results. Common mistakes include failing to document retest periods, using expired primary materials, or skipping proper stability testing. These oversights can result in failed audits and product inconsistencies. Biopharma Consulting Group specializes in creating compliant, science-based Working Reference Standard qualification protocols tailored to your product and process. We help ensure your standards meet regulatory expectations and support consistent, reliable outcomes. Need support with your Working Reference Standard qualification? Visit biopharmaconsultinggroup.com to connect with a biopharma consultant and start building your foundation for success.